Dear viewers, this video is part 3 in this 4-part series on lipid disorders. Efficiency of carbohydrate conversion into fat. During triglyceride synthesis, only about 15% of the original energy in the glucose is lost in the form of heat. The remaining 85% is transferred to the stored triglycerides. Importance of fat synthesis and storage. Fat synthesis from carbohydrates is especially important for two reasons. One, the ability of the different cells of the body to store carbohydrates in the form of glycogen is generally slight. A maximum of only a few hundred grams of glycogen can be stored in the liver, the skeletal muscles, and all other tissues of the body put together. In contrast, many kilograms of fat can be stored in adipose tissue. Therefore, fat synthesis provides a means by which the energy of excess ingested carbohydrates and proteins can be stored for later use. Indeed, the average person has almost 150 times as much energy stored in the form of fat as stored in the form of carbohydrate. 2. Each gram of fat contains almost 2.5 times the calorie of energy contained by each gram of glycogen. Therefore, for a given weight gain, a person can store several times as much energy in the form of fat as in the form of carbohydrate, which is exceedingly important when an animal or human must be highly motile to survive. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Failure to synthesize fats from carbohydrates in the absence of insulin. When insufficient insulin is available, as occurs in persons with serious case of diabetes mellitus, fats are poorly synthesized, if at all. Synthesis of triglycerides from proteins. Many amino acids, that is structural components of proteins, can be converted into acetyl coenzyme A, which can then be synthesized into triglycerides. Therefore, when people have more proteins in their diet than their tissues can use as proteins, a large share of the excess is stored as fat. Regulation of energy release from triglycerides. Carbohydrates are preferred over fats for energy when excess carbohydrates are available. When excess carbohydrates are available in the body, carbohydrates are used preferentially over triglycerides for energy. Thus, an excess of carbohydrates in the diet not only acts as a fat sparer but also increases fat stores. In fact, all the excess carbohydrates not used for energy are stored in the small glycogen deposits of the body or converted to fat for storage. Acceleration of fat utilization for energy in the absence of carbohydrates. All the fat sparing effects of carbohydrates are lost and actually reversed when carbohydrates are not available. The equilibrium shifts in the opposite direction and fat is mobilized from adipose cells and used for energy in place of carbohydrates. Hormonal regulation of fat utilization. Several hormones secreted by the endocrine glands have significant effects on fat utilization. Some important hormonal effects on fat metabolism, in addition to lack of insulin, are probably the most dramatic increase that occurs in fat utilization is that observed during heavy exercise. This increase results almost entirely from release of epinephrine and norepinephrine by the adrenal medullae during exercise as a result of sympathetic stimulation. These two hormones directly activate hormone-sensitive triglyceride lipase which is present in abundance in the fat cells, and this activation causes rapid breakdown of triglycerides and mobilization of fatty acids. Sometimes the free fatty acid concentration in the blood of an exercising person rises as much as eightfold, and the use of these fatty acids by the muscles for energy is correspondingly increased. Thyroid hormone indirectly causes rapid utilization of fat by increasing overall rate of energy metabolism in all cells of the body under the influence of this hormone. Obesity, excess deposition of fat. Obesity is caused by the ingestion of greater amounts of food than can be used by the body for energy. The excess food, whether fats, carbohydrates, or proteins, is then stored almost entirely as fat in the adipose tissue to be used later for energy. The ability of humans to store excess energy in adipose tissue is tremendous, with some people achieving body weights in excess of 500 kilograms, that is 1100 pounds, mainly as a result of fat accumulation. Phospholipids and cholesterol. Phospholipids. The major types of body phospholipids are lecithins, cephalins, and sphingomyelin. Phospholipids are all lipid soluble, transported in lipoproteins, and used throughout the body for various structural purposes such as in cell membranes and intracellular membranes. Formation of phospholipids. Phospholipids are synthesized in essentially all cells of the body, although certain cells have a special ability to form great quantities of them. Their synthesis in humans occurs in the cytosol through endoplasmic reticulum, that is ER membrane located enzymes. Probably 90% of phospholipids are formed in liver cells. Substantial quantities are also formed by the intestinal epithelial cells during lipid absorption from the gut. Eggs, organ and lean meats, fish, shellfish, cereal grains, and oilseeds are the main dietary sources of phospholipids. Specific uses of phospholipids. Phospholipids have several functions, including the following. 1. Phospholipids are an important constituent of lipoproteins in the blood and are essential for formation and function of most of these lipoproteins. In the absence of phospholipids, serious abnormalities of transport of cholesterol and other lipids can occur. 2. Thromboplastin, which is necessary to initiate the clotting process, is composed mainly of one of the cephalins. 3. Large quantities of sphingomyelin are present in the nervous system. This substance acts as an electrical insulator in the myelin sheath around nerve fibers. Phospholipids are donors of phosphate radicals when these radicals are necessary for different chemical reactions in the tissues. 5. One of the most important functions of phospholipids is participation in formation of structural elements, mainly membranes, in cells throughout the body, like cholesterol. Cholesterol. Cholesterol is present in the normal diet. It can be absorbed slowly from the gastrointestinal tract into the intestinal lymph. The main dietary sources of cholesterol are fatty red meat, poultry skin, lard, high-fat dairy products, shellfish, and shrimp. Egg yolks are also rich in cholesterol. Plants make cholesterol in very small amounts. Rather, plants make phytosterols chemically similar to cholesterol. They can compete with cholesterol for absorption in the intestinal tract, thus potentially reducing cholesterol absorption. It is highly fat soluble but only slightly soluble in water. It is specifically capable of forming esters with fatty acids. Indeed, about 70% of the cholesterol in the lipoproteins of the plasma is in the form of cholesterol esters. Formation of cholesterol. Besides the cholesterol absorbed each day from the gastrointestinal tract, which is called exogenous cholesterol. An even greater quantity is formed in the cells of the body, called endogenous cholesterol. Essentially, all the endogenous cholesterol that circulates in the lipoproteins of the plasma is formed by the liver. But all other cells of the body form at least some cholesterol, which is consistent with the fact that many of the membranous structures of all cells are partially composed of this substance. Cholesterol serves as a precursor for the biosynthesis of steroid hormones, bile acids, and vitamin D. Factors that affect plasma cholesterol concentration. Feedback control of body cholesterol. Among the important factors that affect plasma cholesterol concentration are the following. 1. An increase in the amount of cholesterol ingested each day may increase the plasma concentration slightly. The rising concentration of cholesterol provides an intrinsic feedback control system to prevent an excessive increase in plasma cholesterol concentration. As a result, plasma cholesterol concentration usually is not changed upward or downward more than plus or minus 15% by altering the amount of cholesterol in the diet, although the response of individuals differs markedly. 2. A diet high in saturated fat increases blood cholesterol concentration 15 to 25%, especially when this diet is associated with excess weight gain and obesity. This increase in blood cholesterol results from increased fat deposition in the liver, which then provides increased quantities of acetyl coenzyme A in the liver cells for the production of cholesterol. Therefore, to decrease the blood cholesterol concentration, maintaining a normal body weight and a diet low in saturated fat are even more important than maintaining a diet low in cholesterol. 3. Ingestion of fat containing highly unsaturated fatty acids usually depresses the blood cholesterol concentration to a slight to moderate count or amount. 4. Lack of insulin or thyroid hormone increases the blood cholesterol concentration, whereas excess thyroid hormone decreases the concentration of cholesterol. Specific uses of cholesterol in the body. By far, the most abundant non-membranous use of cholesterol in the body is to form cholic acid in the liver. As much as 80% of the cholesterol is converted into cholic acid. Cholic acid is conjugated with other substances to form bile salts, which promote digestion and absorption of fats. A small quantity of cholesterol is used by 1. The adrenal glands to form adrenocortical hormones, 2. The ovaries to form progesterone and estrogen, and 3. The testes to form testosterone. These glands can also synthesize their own steroids and then form hormones from them. A large amount of cholesterol is precipitated in the corneum of the skin, that is the outermost layer of the skin. Cholesterol, along with other lipids, makes the skin highly resistant to the absorption of water-soluble substances and to the action of many chemical agents because cholesterol and the other skin lipids are highly inert to acids and to many solvents that might otherwise easily penetrate the body. Also, these lipid substances help prevent water evaporation from the skin. Without this protection, the amount of evaporation can be 5 to 10 liters per day, as occurs in patients with burns who have lost their skin instead of the usual 300 to 400 milliliters. Cellular structural functions of phospholipids and cholesterol, especially for membranes. The previously mentioned uses of cholesterol and phospholipids are of only minor importance in comparison with their function of forming specialized structures, mainly membranes, in all cells of the body. Large quantities of phospholipids and cholesterol are present in both the cell membrane and the membranes of the internal organelles of all cells. It is also known that the ratio of membrane cholesterol to phospholipids is especially important in determining the fluidity of cell membranes. For membranes to be formed, substances that are not soluble in water must be available. In general, the only substances in the body that are not soluble in water, besides the inorganic substances of bones, are the lipids and some proteins. Thus, the physical integrity of cells everywhere in the body is based mainly on phospholipids, cholesterol, and certain insoluble proteins. Another fact that indicates the importance of phospholipids and cholesterol for the formation of structural elements of the cells is the slow turnover rates of these substances in most non-hepatic tissues, turnover rates measured in months or years. For instance, their function in brain cells to provide memory processes is related mainly to their indestructible physical properties. Dear viewers, this is the end of part 3 in this 4-part series on lipid disorders. For other videos in this series, see Prescribon Medical Education series, Lipid Disorders.